Alright, so in this video, I will be explaining about glycolysis. So basically, I will be concentrating on reactions, regulations, important enzymes, and applied clinical aspects. So let's jump into that. So glycolysis, as you all know, it's a cytoplasmic pathway, and it's basically oxidation of glucose into pyruvate molecule. So that's the glycolysis, and depending on whether oxygen is present or not, so glycolysis can be further divided, means classified into anaerobic glycolysis if oxygen is not available sufficiently, or if the cell do not have mitochondria, or it can be aerobic glycolysis if oxygen is present and mitochondria is present. So anyway, so let's get into the reactions. So very first reaction in glycolysis is a conversion of glucose into glucose 6 phosphate. So I'll just write it as GLU 6 phosphate, glucose 6 phosphate. So this is one of the important reaction, and this is the first irreversible reaction in glycolysis, and the enzyme that catalyzes this reaction is glucokinase and hexokinase. So we are going to talk about glucokinase enzyme and also hexokinase enzyme. So note that it is a single reaction, that is glucose is converted into glucose 6 phosphate, but it will be done by two enzymes, and these are individual enzymes and they conduct individual reactions, but we'll have to look into why one reaction here is it needs like two enzymes. Why hexokinase and glucokinase one when it could have been done by single enzyme. Anyway, so I'm going to go into details of that later. So conversion of glucose into glucose 6 phosphate, you need ATP here. One energy phosphate is required. ATP will be converted into ADP molecule. So that's the very first important reaction in glycolysis. Now let's move on to see like what is the next important reaction in glycolysis. I will be concentrating only on the important reaction, and other reactions I don't go into details. Now actually what happens is glucose, which when it is converted into glucose 6 phosphate, so glucose 6 phosphate will be undergoing a reversible reaction to become fructose 6 phosphate. Now the fructose 6 phosphate, it will break into fructose 1,6 bisphosphate, fructose 1,6 bisphosphate. So that's the third reaction, and that is a irreversible reaction. Third reaction is irreversible reaction, and also in this reaction there is a requirement of ATP again. So one more ATP is needed to make sure that this reaction is going on in a forward direction. So ATP is broken down into ADP. So there is consumption of one ATP there. So this particular reaction, it will be catalyzed by an enzyme called phosphofructokinase. Phosphofructokinase enzyme, and that number one, phosphofructokinase enzyme number one, which is simply written as PFK1 enzyme. So this is the enzyme that will catalyze conversion of fructose 6 phosphate into fructose 1,6 bisphosphate. This is another important enzyme that we need to go into details. So I'm going to take you into the enzyme importance and all that. So fructose 1,6 bisphosphate will be converted to basically it will be broken down into glyceraldehyde 3 phosphate, glyceraldehyde. I'll just write GLY, glyceraldehyde 3 phosphate, and also it will be converted to another molecule. It's called DHAP, so dihydroxyacetone phosphate. So these are two molecules that are coming out of fructose 1,6 bisphosphate, and this particular reaction is catalyzed by an enzyme called aldolase enzyme, which belongs to lyase class of enzymes. So not really that important, but since like lyase class, it belongs to lyase class of enzyme. It is an example for lyase enzymes. So I'll write it here. Aldolase enzyme is a lyase class of enzyme. It breaks a molecule without adding water, unlike hydrolase enzymes. Anyway, so glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they can undergo interconversion. So there can be interconversion of these two molecules by triose phosphate isomerase enzyme, not really that important. So overall, what we get for our easier understanding, and also when energy requirement is there for a cell, so it is going to convert DHAP back into glyceraldehyde 3-phosphate. Overall, what happens is we are going to have two molecules of glyceraldehyde 3-phosphate here, because assuming that DHAP will be converted back into glyceraldehyde 3-phosphate. So let me write it as like two molecules of glyceraldehyde 3-phosphate coming from one molecule of glucose, because glucose is a 6-carbon molecule and glyceraldehyde 3-phosphate is a 3-carbon molecule, and we have two molecules of glyceraldehyde 3-phosphate. That means total we have 6 carbon in the form of 2 glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate, from here onwards all the reactions you need to know, like multiply by 2, so times 2 times basically. So glyceraldehyde 3-phosphate will undergo oxidation reduction reaction, reversible one, and it will become 1,3 bisphosphoglycerate, 1,3 BPG, 1,3 bisphosphoglycerate. So 2 glyceraldehyde 3-phosphate, two molecules, I will be converted to two molecules of 1,3 bisphosphoglycerate. So this particular reaction, it will be catalyzed by enzyme glyceraldehyde 3-phosphate dehydrogenase. So glyceraldehyde 3-phosphate dehydrogenase. So dehydrogenases, they belong to oxidoreductase class of enzyme. That means there will be oxidation and reduction reaction going on in this kind of reactions. So here what happens, glyceraldehyde 3-phosphate is oxidized into 1,3 bisphosphoglycerate. In this particular reaction, NAD, oxidized molecule NAD will be reduced into NADH plus H plus molecule. And also in this reaction, so there will be incorporation of inorganic phosphate molecule. So the Pi, inorganic phosphate, will get into the reaction, and that's what is the additional phosphate that you get, glyceraldehyde 3-phosphate. You're going to get 1,3 bisphosphoglycerate. So there is an additional phosphate here in the first carbon, and that phosphate is basically coming from inorganic phosphate from the cytoplasmic inorganic phosphate pool. So anyway, now 1,3 bisphosphoglycerate, it is a energy molecule, kind is basically one of the energy molecule that contains high energy phosphate bond. So what happens in 1,3 bisphosphoglycerate will undergo dephosphorylation process and become 3-phosphoglycerate. So 1,3 bisphosphoglycerate becomes 3-phosphoglycerate. I'll just write it as 3PG. PG means phosphoglycerate, 3-phosphoglycerate. So during this process, there will be generation of ATP. That's one of the important step here. So the ADP will undergo phosphorylation. ADP will undergo phosphorylation in the reaction and become ATP, adenosine triphosphate. So this particular reaction, it will be catalyzed by an enzyme called phosphoglycerate kinase. Phosphoglycerate kinase. 1,3 bisphosphoglycerate converting into 3-phosphoglycerate. It will be done by phosphoglycerate kinase enzyme. As the name says, kinase. Kinases are transferases. So what they do, they are transferring the phosphate present in the first carbon to ADP, and that's when ADP is converted to ATP molecule. And now ATP is a high energy molecule, and that ATP is synthesized in this particular reaction. So in the first and third reactions, see, this is the first reaction, this is the third reaction. So first and third reaction, there were consumption of ATPs there. Actually ATPs are consumed and ADPs were released, whereas here in this reaction, actually ATP is synthesized in the cytoplasm itself, because all these reactions are going on in the cytoplasm. So there is generation of ATP in the cytoplasm, and that's why this is an example for substrate level phosphorylation. So Phosphoglycerate kinase is an example for substrate level phosphorylation reaction. That's one of the example here in glycolysis. Now let's see what happens to 3-phosphoglycerate. Now the 3-phosphoglycerate will undergo reversible reaction to become 2-phosphoglycerate. Not much importance here. Now the 2-phosphoglycerate will be undergoing another reversible reaction to become phosphoenolpyruvate. So phosphoenolpyruvate. Now the phosphoenolpyruvate, you can simply write it as PEP, phosphoenolpyruvate. So this particular reaction, it will be catalyzed by enzyme enolase. Name of the enzyme here is enolase. So enolase enzyme is the one that will catalyze conversion of 2-phosphoglycerate into phosphoenolpyruvate. What is the importance of enolase? Enolase enzyme, it will be inhibited by fluoride. So that's why, like, fluoride present in the water, or maybe fluoride present in the amount of fluoride that we add in toothpaste or oral anti-cavity solution, it will be just sufficient to inhibit actually oral bacterial enolase enzyme, like the bacteria present in our oral cavity. So they also survive by conducting glycolysis. So it now fluoride in the toothpaste inhibit bacterial enolase, unlike human enolase. Human enolases are not inhibited. Bacterial enolase is inhibited. That means glycolysis is inhibited. That's why bacteria won't be able to generate sufficient ATPs, and thereby they won't be able to multiply. So that's how bacterial inhibition can be done to prevent oral cavities, basically, or dental caries. So that's why fluoride is added in the toothpaste or oral anti-cavity solution. Now coming to phosphoenolpyruvate converted into pyruvate molecule, another irreversible reaction in glycolysis. So product is pyruvate, and the enzyme that converts phosphoenolpyruvate to pyruvate, it is pyruvate kinase enzyme. Now this speciality of this reaction is, it is another example for substrate level phosphorylation, because in this reaction ADP is phosphorylated to make ATP. Let me change the color, because we are making an ATP here, unlike consumption of ATP. So there is formation of ATP in this reaction. So ATPs are formed. So this is a high energy phosphate that is formed here, and that's why pyruvate kinase is another example for substrate level phosphorylation process. So we got pyruvate now. Now what will happen to this pyruvate that is generated by pyruvate kinase, which is an irreversible reaction? Now the pyruvate has got two options. Pyruvate can enter into mitochondria. So if mitochondria is present, oxygen is present, so pyruvate can basically enter into mitochondria by pyruvate transporter, and it will undergo aerobic glycolysis. So basically aerobic glycolysis will go on. What if cell do not have mitochondria? What if, like, oxygen availability is less? At that time pyruvate, it will be diverted into lactate formation. Pyruvate will be converted into lactate. Okay, and this particular reaction is catalyzed by lactate dehydrogenase enzyme. Simply I will write it as LDH here. So lactate dehydrogenase. So lactate dehydrogenase is the one that will convert pyruvate into lactate molecule. So let me rewrite lactate dehydrogenase, LDH, lactate dehydrogenase. Okay, so during this process, what happens is, because the pyruvate is undergoing reduction process, so that means there must be a molecule that undergoes oxidation, and that is the molecule that undergoes oxidation here is NADH plus H plus molecule is oxidized into NAD plus molecule, NAD. NADH plus H plus is oxidized into NAD, while pyruvate is reduced into lactate by lactate dehydrogenase. How do you get NADH H plus? So, and why pyruvate has to convert into lactate in anaerobic glycolysis? So, this is anaerobic glycolysis, anaerobic glycolysis, where pyruvate is converted to lactate. So, why this happens? Why pyruvate has to be converted into lactate? It is only because, so in the glycolysis, there is a step that needs NADHH plus, NAD plus molecule. So here is the molecule. So this one, NAD plus molecule, is needed for glyceraldehyde 3 phosphate. So glyceraldehyde 3 phosphate dehydrogenase enzyme, glyceraldehyde 3 phosphate dehydrogenase, is the one that needs NAD plus, and this glyceraldehyde 3 phosphate dehydrogenase, it needs NAD plus. As you can see, NAD plus is needed for this reaction, where NAD plus is converted to NADH plus H plus. So we need to continuously supply this NAD plus molecule so that glycolysis actually continues to make pyruvate, and pyruvate has to go into mitochondria or has to go into lactate formation. So the only reason why we convert pyruvate into lactate is to oxidize NADHH plus and to give this NAD plus molecule, because this NAD plus, which is produced in lactate dehydrogenase reaction, it can be used in glyceraldehyde 3 phosphate reaction and make NADHH plus molecule, and that NADHH plus molecule can undergo basic oxidation in lactate dehydrogenase, and the plan here is to continuously supply NAD plus molecule. So the importance here is to supply NAD plus molecule. That is why there is a formation of lactate here. So we want this NAD plus molecule, because this NAD plus molecule, it is needed by glyceraldehyde 3 phosphate dehydrogenase. Thereby glucose can undergo continuously oxidation, and NADHH plus that is made in glyceraldehyde 3 phosphate dehydrogenase, it will be used by lactate dehydrogenase enzyme. So this is the plan in anaerobic glycolysis. That's why during anaerobic glycolysis there is always an reduction of pyruvate into lactate, while NADHH plus is oxidized to NAD plus to continuously supply NAD plus molecule. That's what happens in the red blood cell. So the RBCs, they conduct anaerobic glycolysis, and also skeletal muscle under vigorous activity, strenuous physical activity, whenever respiratory chain or electron transport chain is kind of getting saturated, when the oxygen is the only limiting factor. But still, if there is a requirement of energy for this exercising skeletal muscle, so energy has to be continuously supplied, and that's why pyruvate is diverted into lactate in exercising skeletal muscle or in the red blood cell. Only reason for that is to regenerate NAD plus in the cytoplasm, because no mitochondria is present here in the red blood cell. So regeneration of NAD plus is important to carry out glycolysis, because it is necessary in glyceraldehyde 3 phosphate dehydrogenase. Because every time glyceraldehyde 3 phosphate dehydrogenase consumes NAD plus and make NADHH plus, and that will be eventually oxidized by lactate dehydrogenase. So this is why in exercise in skeletal muscle, so the lactate, it can build up in the skeletal muscle, and it can lead to, eventually can lead to pain. Anyway, so this is about aerobic and anaerobic glycolysis. So I told you that I'll come back to glucokinase, hexokinase step and some of the important steps. So let me explain you some of those steps in brief. So let's move on to see where I was explaining glycolysis. So now look at the energetics of glycolysis. So 1 ATP is consumed in this step, first step. Third step, another ATP is consumed here. So 2 ATPs are consumed so far 2 ATPs are consumed. So I'll write it as minus 2 ATPs, minus 2 ATPs. And now let's see how many ATPs are actually produced. So as we come here, so I told you, like glyceraldehyde 3 phosphate 2 molecules. So every reaction from here onwards will be 2 2. So although I forgot to write, but I told you, so these are all like 2 2 number. All reactions in, as we go on from glyceraldehyde 3 phosphate onwards, they'll be always 2 2 number. Anyway, so you get one NADHH plus in this particular reaction, and if it is an anaerobic glycolysis, so this NADHH plus will be used by lactate dehydrogenase. Effectively, it do not contribute to any ATP levels under anaerobic conditions. So we have taken that out. And then there are two substrate level phosphorylations. See, there is a phosphoglycerate kinase, one substrate level phosphorylation. So two molecules of 1,3-bisphosphoglycerate, when it is converted to two molecules of 3-phosphoglycerate, you get two ATPs. So remember those two ATPs there. Similarly, in pyruvate kinase reaction, two phosphoenolpyruvate converting into two pyruvate, you get two more ATPs. So total we have four ATPs being produced there. So four ATPs produced, and two ATPs are actually used in the initial first and third reaction. So overall, so you get net 2 ATP. So this is the net ATP yield, yeah, under anaerobic glycolysis. So under anaerobic glycolysis, so the net ATP yield is 2 ATP. So from where those 2 ATP comes? Actually, those 2 ATPs will come from pyruvate kinase reaction, because like first reaction and the third reaction, first and third reaction, 2 ATPs consumed by for, and phosphoglycerate kinase step, 2 ATPs you make, so 2 minus 2 0 there, but the net gain actually it will come from pyruvate kinase step. So that's what is these two ATPs. So what happens if there is a pyruvate kinase deficiency? So these two ATPs do not come, and that's why it leads to overall lack of ATP in pyruvate kinase deficiency, especially in the red blood cell, and that is why hemolytic anemia can happen. Now let's see how many net number of ATPs you get under aerobic glycolysis. So let's move on to see aerobic glycolysis. So aerobic glycolysis is oxidation of glucose into 2-pyruvate in the presence of mitochondria and sufficient oxygen. Now calculate that. So 2 ATPs are initially consumed, first and third reaction 2 ATPs consumed, and phosphoglycerate kinase 2 ATPs, pyruvate kinase 2 ATPs. So total 4 ATPs you got there, and also additional ATPs, 2 NADH, 2 molecules have NADHH plus here. So because in this step, so you got 2 NADH plus 2 H plus molecule, and these 2 NADH plus molecules, once they go into electron transport chain, because mitochondria is sufficiently present, so 1 NADH plus is 2.5, so 2.5 plus 2.5, so total you get 5 ATPs from 2 NADH plus. So those 5 ATPs, so 4 plus 5 ATP there. So that is 9. 9 minus 2, 2 ATPs are consumed. So you get 7 ATPs, actually 7 ATPs. So in aerobic glycolysis you get total 7 ATPs. Under anaerobic glycolysis you get net 2 ATPs. This is the energy yield from glycolysis. I have a video on energy yield glucokinase and glucokinase reaction. So, conversion of glucose into glucose 6 phosphate catalyzed by glucokinase and hexokinase. So why do we have two enzymes for doing the same thing? So you need to so in order to understand that, we need to know the Km and Vmax of glucokinase and hexokinase enzymes. So I'm going to draw the glucokinase hexokinase reaction velocity curve. So this is for glucokinase. Let's write it as glucokinase. And for hexokinase, so here is the curve for hexokinase enzyme. Okay, now let's take the Km and Vmax of these two enzymes. So here is this is the Vmax for glucokinase, Vmax of glucokinase, and Vmax for this is hexokinase here, hexokinase. So Vmax for hexokinase is here. Now take the half of the Vmax of glucokinase. So half of the Vmax for glucokinase, let me change the color here. Say it is the half of the Vmax for glucokinase. So it will be, say, somewhere here. Say let's write it as 5 moles or 5 millimoles here. And for hexokinase, so half of the Vmax for, so this is the Vmax for hexokinase, and take half of it, and this will be 0.05 millimoles. Now the hexokinase has got low Km, so this is a low Km enzyme. Hexokinase is a low Km enzyme, whereas glucokinase is a high Km enzyme. So as you all know, lower the Km, higher the affinity. That means hexokinase has got higher affinity for glucose compared to glucokinase, which has got lower affinity for glucose. So what is the meaning of this? So any enzyme that has got low Km, that means having higher affinity, they will be able to work even under low blood glucose concentration, like even under hypoglycemic concentration. So hexokinase is present in the peripheral tissues. Like basically hexokinase is present in peripheral tissue, except in the liver and beta cells of pancreas. This is where you don't find hexokinase. Other than these two tissues, all the other tissues have got hexokinase, and they really have low Km for glucose. That means they are always working and they are always saturated with the glucose. Whereas the glucokinase having high Km, having lower affinity, so it works only when there is high blood glucose level, like after food. So glucokinase is present in the liver and beta cells. So basically liver has to take up glucose only when it is available plenty. The reason for this is it has to leave that glucose under low blood glucose concentration for other tissues to use it. And beta cells, why it has glucokinase there? Because beta cells, they are responding to glucose, high glucose, and release insulin. That means only insulin has to be released when there is a high blood glucose concentration. It should not be released whenever there is a low blood glucose concentration. That's the reason why glucokinase having high Km is located in the beta cells of pancreas, to make sure the beta cell is releasing insulin only when there is high blood glucose. And why it is present in liver? Because to make sure liver is using glucose only when there is high glucose available in the blood. And also take a note of Vmax of glucokinase and hexokinase. So the glucokinase is a high Vmax enzyme, just like Km. It also has high Vmax, whereas hexokinase is a low Vmax enzyme. So hexokinase is low Km and low Vmax enzyme, and glucokinase is high Km, high Vmax enzyme. So hexokinase having low Vmax, it will make sure that tissues will use only that much amount of glucose, whatever it is necessary for the tissue, because it saturates early, and any glucose that is not yet phosphorylated into glucose 6-phosphate, it will simply go out of the tissue by the glucose transporter into the blood, and that glucose can be used by other tissues which needs it. So having low Vmax, it will make sure that tissues will use only the amount of glucose that they need, and anything in excess of their need will be sent out into the blood. So that is good for peripheral tissues, because so that they can use glucose only as per their needs, and so that other tissues can also get glucose for their own metabolic needs. And whenever there is high blood glucose level, so glucokinase having high Km and high Vmax. Vmax comes it indicates the capacity of an enzyme. So glucokinase having high Vmax, it is a high capacity enzyme. It means it can go on converting glucose into glucose 6-phosphate to the maximum amount, and make sure that glucose do not exceed in the blood. So overall, having IVMAX, glucokinase is going to prevent a person going into hyperglycemia under fed condition. Having hexokinase low Vmax, it will make sure that person maintains the blood glucose level, and that means a patient do not go into hypoglycemia. Because imagine if the hexokinase has got high Vmax like glucokinase, it would have, all the tissues would have consumed more and more glucose, and soon blood glucose would have been low, and person might have go into hypoglycemia. So low Vmax, hexokinase having low Vmax will prevent from hypoglycemia. Glucokinase having high Vmax will prevent person going into hyperglycemia under fed condition, because having a high capacity, it is able to use more glucose. That's the importance of glucokinase and hexokinase steps. That's why we have two enzymes for the same reaction. Now let's the first slide. I want to see our next important enzyme that I said I'll explain is conversion of fructose 6-phosphate into fructose 1,6 bisphosphate. This reaction will be catalyzed by enzyme PFK1 enzyme, phosphofructokinase 1 enzyme. Now this phosphofructokinase 1 enzyme, especially in the peripheral tissue, it will be positively modulated by AMP. AMP is a positive modulator on phosphofructokinase 1 enzyme. Thereby more and more glycolysis will go on, and AMP is an energy hunger signal in a cell. A lot of AMP means increased AMP means lack of ATPs. That means glycolysis has to go on sufficiently. Now there are two negative modulators on PFK1. One is ATP is a negative modulator, and citrate in the cytoplasm is a negative modulator on PFK1. These are negative modulators. Whenever there is sufficient ATPs, whenever there is increase in citrate, so these are energy rich molecules, so they basically decrease glycolysis pace by acting negatively on PFK1. Now there is another positive modulator, especially in the liver, in the hepatocyte. The concentration of fructose 2,6 bisphosphate, fructose 2,6 BP, and this fructose 2,6 bisphosphate concentration, whenever it is high, it will have a positive effect on PFK1. So depending on the concentration of fructose 2,6 bisphosphate, so the glycolysis pace can be altered. So whenever we are in fed condition, so the insulin will have a positive effect on enzyme called PFK2, phosphofructokinase 2 enzyme. It positively modulates phosphofructokinase 2 enzyme and make sure fructose 2,6 bisphosphate is synthesized by PFK2 enzyme. That thereby it will have a positive effect on PFK1, and thereby glycolysis will increase. So insulin indirectly it increases glycolysis by keeping PFK2 enzyme active. Thereby PFK2 makes fructose 2,6 bisphosphate. Thereby fructose 2,6 bisphosphate will have a positive effect on PFK1 enzyme. Now there is another, like, glucagon. Whenever we are in fasting conditions, so glucagon is the fasting hormone, and what this glucagon does. So glucagon will have a negative effect on PFK2 enzyme. Glucagon will have a negative effect on PFK2 enzyme. Basically what it does, it's going to decrease the kinase part of PFK2 enzyme and increases phosphatase part of PFK2 enzyme, and thereby what happens is, so it's basically glucagon is going to activate PFK2 phosphatase enzyme. And this PFK2 phosphatase, what it does, it is going to decrease concentration of fructose 2,6 BP. Fructose 2,6 BP concentration will be decreased by glucagon by keeping PFK2 phosphatase component. Thereby fructose 2,6 bisphosphate is converted to fructose 6 phosphate. Thereby overall fructose 2,6 BP concentration decreases, and that means its effect on PFK1 is not there. So glucagon indirectly it decreases PFK1 activity by decreasing fructose 2,6 BP, which is a positive modulator on PFK1. And when the positive modulator availability is less, so PFK1 is also decreasing activity, thereby overall decreases glycolysis. I have a separate video on this, so link for that video I have given in the description below. So you can watch that PFK1 PFK2 regulation by insulin and glucagon. It's a very detailed video. I'm sure you're going to that video will be helpful to you. Now let's move on to see our third important reaction, that is conversion of glyceraldehyde 3 phosphate into 1,3 bisphosphoglycerate. So this is the reversible reaction, and in this reaction, as I already told you, NAD plus is needed for this reaction. NADHH plus goes out of the reaction. Inorganic phosphate, as it gets into the reaction to make 1,3 bisphosphoglycerate, catalyzed by glyceraldehyde 3 phosphate dehydrogenase enzyme. Now, one of the importance of this, apart from like no, it is a reaction that makes NADH plus H plus and all that, and of course it needs NAD plus. Now there is a molecule like no heavy metal ion arsenate. Arsenate, what it does, it's going to go and bind to a site in the glyceraldehyde 3 phosphate where inorganic phosphate is binding. So in the presence of glyceraldehyde 3 phosphate is direct, it's going to bind the glyceraldehyde 3 phosphate dehydrogenase. And arsenate, what it does, it directly converts glyceraldehyde 3 phosphate into 1 3 into 3 phosphoglycerate, because 1 3 bisphosphoglycerate, it will be converted to 3 phosphoglycerate normally, and during that process ATP will be generated as we have seen before. So ATP, this is one of the example for substrate level phosphorylation by phosphoglycerate kinase enzyme. So what happens in the presence of arsenate is glyceraldehyde 3 phosphate, it directly converts 1 3 bisphosphoglycerate into 3 phosphoglycerate without making 1 3 bisphosphoglycerate, without making ATP by phosphoglycerate kinase. Overall, it bypasses the reaction. There is no NADHH plus formed, no 1 3 bisphosphoglycerate formed, no ATP formed. So entire glyceraldehyde 3 phosphate in the presence of arsenate, it will be converted into 3 phosphoglycerate. So overall, what it does, it's going to bypass the reaction and still continue glycolysis, but cell won't be able to make ATP in substrate level phosphorylation. Cell won't be able to make NADHH plus, because if this NADHH plus goes to electron transport chain, it would have given like 2.5 ATPs in the electron transport chain. So this is what is the effect of arsenate on glyceraldehyde 3 phosphate by bypassing the reaction. Now, and also I have other important functions of the important to it is actions that in glycolysis is say substrate level phosphorylation. Substrate level phosphorylation, meaning of this is phosphorylation is going on in the cytoplasm itself, like substrate level phosphorylation, that is ATP generation. ATP generation normally it happens in the mitochondria, whereas in substrate level phosphorylation, ATP generation is going on in the cytoplasm itself. This is something special. And there are two reactions in glycolysis that can catalyze substrate level phosphorylation, and that is phosphoglycerate kinase enzyme. Phosphoglycerate kinase. Make sure you repeatedly write these enzyme names or say these enzyme names so that you are familiar with the enzymes. And also important reactions, I recommend you to remember substrate and the product for each of those reaction enzymes. So there are two substrate level phosphorylation in glycolysis. One is phosphoglycerate kinase and other is pyruvate kinase enzyme, pyruvate kinase. So this is another example for substrate level phosphorylation. Just to remind you, like what is the substrate and product for phosphoglycerate kinase. So the phosphoglycerate kinase substrate is 1,3 bisphosphoglycerate, which will be converted to 3 phosphoglycerate. For pyruvate kinase, it is 2 phosphoglycerate is converted to phosphoenolpyruvate. This is the product. So these are the example for substrate level phosphorylation. Now let's move on to see our next important reaction slide here. So I have written something wrong. So let me correct that here. So pyruvate kinase enzyme, what it does, it's going to convert phosphoenolpyruvate into pyruvate molecule. So that's what is the job of pyruvate kinase. Let me correct that here. Pyruvate kinase converts phosphoenolpyruvate into pyruvate molecule. So that's what is the job of pyruvate kinase, and that's an example for substrate level phosphorylation. So let me take detailed in glycolysis, and that is enolase enzyme. So enolase enzyme, what it does, it's going to convert 2 phosphoglycerate into phosphoenolpyruvate. I'm sorry, I'll have to take you back to my previous, get back to our this slide here. So I was explaining 2-phosphoglycerate into phosphoenolpyruvate. It will be done by enzyme enolase enzyme. So I was saying that fluoride, that is usually as a sodium fluoride. So fluoride, which is added in anti-cavity solution or toothpaste, fluoride in toothpaste or anti-cavity solution. So sodium fluoride is added. So amount of fluoride that is added in this toothpaste or anti-cavity solution, so it is just sufficient to inhibit bacterial enolase enzyme. So it inhibits only enolase bacterial enolase enzyme is inhibited, because that's the amount of fluoride which is added here. So why bacterial enolase? Because the oral cavity has got like billions of bacteria, and what they will do, they survive on the glucose that is coming from human, and they will conduct anaerobic glycolysis. You know, the product of anaerobic glycolysis is lactate, and this lactate, it decreases the local pH, make it acidic, and thereby lactate will degrade enamel, which is the outer part of the teeth, and that's how the dental cavities will form, dental caries or cavities. So reason for that is anaerobic glycolysis conducted by the bacteria, anaerobic glycolysis, and the product of anaerobic glycolysis is lactate, and lactate by decreasing the pH and basically leads to dental cavities and caries. So by using fluoride and inhibiting bacterial enolase, overall you inhibit glycolysis in the bacteria, and thereby they won't be able to multiply properly, and that's the mechanism of fluoride acting on enolase enzyme. Remember, 2-phosphoglycerate is converted to phosphoenolpyruvate by enolase, which is blocked by fluoride. Now let's move on to see our the last and that video. So and one but last. So because I would like to still tell about lactate dehydrogenase importance again. So the last but one is conversion of phosphoenolpyruvate into pyruvate molecule. So this will be done by pyruvate kinase enzyme, pyruvate kinase, and of course this is an example for substrate level phosphorylation that I have already explained you. ADP is converted to ATP in this reaction. So overall, this is an energy yielding step, and in glycolysis, the first and third step is the step where there will be ATPs are consumed, and later on we are going to get ATPs. And so if there is any deficiency of pyruvate kinase enzyme, imagine there is a mutation in a gene coding for pyruvate kinase enzyme. So the pyruvate kinase deficiency is happening. Pyruvate kinase deficiency. Whenever there is a pyruvate kinase deficiency, so especially if this happens in the red blood cell RBCs, because red blood cells, they conduct anaerobic glycolysis. So when there is anaerobic glycolysis, so in anaerobic glycolysis, red blood cells are effectively, they will be making 2 ATPs per glucose molecules. For every glucose which is oxidized into 2 lactate molecules, there will be generation of only 2 ATPs, and these 2 ATPs are the ones that will support red blood cell membrane integrity and sodium potassium pump and all that. So whenever there is a deficiency of pyruvate kinase enzyme, so the red blood cell membrane, so it kind of breaks open. So there will be, because of the lack of ATP, membrane integrity won't be proper. So the red blood cells, they start leaking hemoglobin molecule. So hemoglobin molecule will come out of the red blood cell, and this hemoglobin, once it comes out of the red blood cell, so it will be broken down into bilirubin molecule. So this bilirubin will increase. So there will be increase in bilirubin, and that will give rise to jaundice. And also, as the hemoglobin comes out and being destroyed, so there will be decrease in hemoglobin content. So overall, hemoglobin levels will be decreased in the blood. So decrease in blood hemoglobin levels. So in the blood, hemoglobin is decreased, and that we call it as anemia. So that's why Pyruvate kinase deficiency is the most common cause for hemolytic anemia in glycolysis. So hemolytic anemia, basically patient will have hemolytic anemia. So pyruvate kinase deficiency is the most common glycolytic enzyme deficiency that leads to hemolytic anemia. So patient will have jaundice and lack of hemoglobin, and putting that patient into all the signs and symptoms of lack of hemoglobin. Anyway, so why only two ATPs are coming from anaerobic glycolysis, and how many ATPs actually come under aerobic glycolysis? In order to, so if you have any doubt, please watch that video and understand what is the net ATP yield from glycolysis. So this is what is all about glycolysis steps. So I told you I will be explaining you about reactions, um, and then regulation of glycolysis actually goes on predominantly on PFK1, although pyruvate kinase can be regulated by insulin positively and glucagon negatively. And applied aspect, so applied clinical aspect, I have already explained you about arsenate, fluoride, and hemolytic anemia. So that's all about glycolysis. So thanks for watching, and if you want to get notification whenever I make a new video, almost like I started to make videos like one or two videos, or maybe 3 to 4 videos per week. So a lot of videos will be coming up on new topics. So you can please consider subscribing to this channel so that you get, and put the bell button on, like no the notification on, to get notification there. And also if you like the video, so please give thumbs up, and also if you have any questions, so you can comment or leave feedback on the video. So I appreciate that. Thanks for watching, and I will see you in my next video. Till then, you take care.